Hello everyone, welcome back to Dr. K. Primer lecture series presenting by Dr. K. Prem, that's me. Today we will talk about most well-known cloning vector that is pUC18. pUC18. So you all aware of the pUC18, right? So that's a cloning vector. So you all, what is? Let us look into the nomenclature of pUC18. P stands for plasmid. That's always in the any plasmid name. If you see, the P is in the lower case. Lower case. Then here the UC stands for University of California. This vector is developed by University of California. Then 18 is a variants in the PUC series. So there is a pUC8, pUC9, pUC90, 18, and pUC19. So that's a variant vectors in the PUC series. And here I'm saying it's a cloning vector. Vectors are two categories. One is a cloning vector and second one is an expression vector. Cloning vectors are used to amplify the massive amplification of cloned gene in vivo. Whenever you clone your gene of interest into the cloning vector, that's a high copy number plasmid. It will amplify massively and get a more plasmid. Along with the plasmid, your insert also amplified, and you get a that your insert is amplified. So whatever purpose, downstream process purposes, maybe preparation or subcloning, you want to do the subcloning from cloning vector to expression vector where you need more amount of your gene, or if in case of sequencing. So that's why always people starts with the cloning in the cloning vector, then they'll move to the expression vector. In this lecture, I'm going to talk about features of pUC18 or elements of pUC18 and their functions, and what are the advantages of pUC18 being a cloning vector over the PBR 322 that is we discussed in the previous lecture, right? All right, let's move to the elements of pUC18. So as you all know that plasmids are covalently closed circular extra chromosomal double strand DNA molecules, right? You see, this is a covalently closed, means there is no open end, nicks are not there. So covalently closed circle, it's a closed one. And circular extra chromosomal, right? DNA molecule, double strand DNA molecule, that they're called as a plasmid. This is an artificial plasmid. pUC18, pUC18. The size of this vector is 2686 base space. So smallest vector for cloning purpose. So ideal cloning vector should have two features. One of them is small size. Small size allows us to clone the bigger fragments. So first the feature is small size of the vector is the ideal feature of a cloning vector, and this is pUC18 fulfill that first criteria. Here the pUC18 have a PLAC, that's a promoter. You all know promoters, right? Promoters are the sites for binding with the RNA polymerase, and then once they binds to the promoter, they initiates the transcription. So this PLAC promoter is isolated from lac operon of E. coli. You all know lac operon, right? So lac operon is having three genes, structural genes. One is a beta galactosidase and transferase and transporter, right? So they are in the operon. So the lac operon is on whenever there is a lactose and whenever there is a absence of glucose, right? So here the lac promoter is isolated from the that operon and inserted into the pUC18 vector. And here lac promoter, PLAC promoter is going to be induced by the allolactose. Allolactose, that is nothing but IPTG. That's an inducer. It will bind to the PLAC and allows, it will bind to the repressor of the lacoperon and allows the binding of RNA polymerase to the PLAC and drives the transcription. So IPTG is a IPT is a isopropyl beta galactoside. That's a non-metabolic allolactose. Means it is not metabolized. It cannot be metabolized by the system. Whatever amount you add to the system, it will be there forever, the same concentration. Hence, IPTG is used to switch on the lac promoter, right? Under the downstream of the lac promoter, there is a small gene that's called as LAC-Z prime. LAC-Z prime. So this is LAC-Z prime is a shortest or short fragment of beta galactosidase gene. So as you all know that beta galactosidase enzyme, which breaks down the lactose into glucose and galactose, right? This is having two domains. N-terminal domain is called as alpha peptide, and C-terminal domain is called as omega peptide. So the alpha fragment is from 1 to 140 amino acid, and the rest is almost like 900 amino acid is the omega. If you chemically break down this enzyme into alpha and beta, alpha and omega fragment, and then you again club them, they become a functional protein. So here in this pUC18 or pUC18, LAC-Z, that's a 30 nucleotides encoding gene, that's a LAC-Z prime. So LAC-Z prime is inserted downstream to the promoter. So whenever is IPTG binds to the repressor, RNA polymerase binds to the lac promoter and drives the expression of LAC-Z prime, which encodes the alpha peptide. And this alpha peptide and omega peptide in the host gene. That's in the host gene, omega from the host gene. And when you transform into the host, E. coli is having the omega fragment and alpha fragment from the vector and omega fragment from the host, and they'll club and associate and form a functional beta galactosidase, right? And gives the blue color colonies in the presence of X dot. That's I'll come to the next one. So this LAC-Z prime is encodes the alpha fragment of beta galactosidase. In that, so that's a 30 amino acid of and beta galactosidase. And in the LAC-Z prime region, there is a short region called as MCS, multiple cloning site or polylinker. So these sites provides the more number of restriction sites. I can tell you, you see, there is a eco R1, SST1, SKP1, SMA1, BAMH1, SBA1, SAL1, and you see PST1, SPH1, and IND3, so many. Almost 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We have a 10 restriction endonucleases in the lag Z prime region that's inserted without losing the open reading frame of lag Z prime. So which allows the cloning of variety of DNA fragments. Sometimes what happens, your DNA fragment is having a small site. You cannot use the SMA. And if you have a eCoron, you cannot use, but you can use the SBA, SOL1, or End3, or BAMH1. So it is 10 different restriction sites are there in the MCS or Polylinker, which will allows us to clone our DNA fragment very easily. And also, because of MCCS, we can clone our gene of interest in a direction oriented. For example, your insert, if you're having a EcoR1 at the promoter region, at the starting codon, right? And the end codon is you are taking the end 3, right? So exactly the start codon comes down under the P-LAC promoter, and the end codon will goes to the end of this LAC cell, right? So directional cloning is also possible with the multiple cloning sites, right? So you need to use the 2 restriction enzymes to clone the your gene of interest, that is called as directional cloning. The next element is ampicillin resistance marker. Ampicillin resistance marker. What is else? That's a genetic selection marker. So to score the transformants, when you transform your vector and insert or into the host, the host which received the plasmid is resistant to one of the antibiotics because the your plasmid is carrying a antibiotic resistance gene. Here, AP stands for ampicillin resistance gene. What is that ampicillin resistance gene? It's nothing but beta lactamase. Beta lactamase. Beta lactamase is useful in the selection of transformants. So whenever vector or plasmid enter into the E. coli, that E. coli become resistant to the ampicillin, then they can grow in the presence of ampicillin. So the beta lactamase converts the ampicillins or belongs to the penicillin family, break down the beta lactam ring of penicillins and converts into the penicillinic acid, which is biologically inactive. So the genetic selection marker, ampicillin resistant gene, that is beta lactamase, is there in the pUC18 to screen the transformants, right? The last one, not the least one, that's origin of replication. ORI stands for origin of replication. So origin of replication is very, very essential element of the plasmid as well as the cloning vehicle, because that is enable the vector to be replicated autonomously or independently. So autonomous replication or independent replication of the cloning vehicle is all depends on the presence of origin of replication, from where the replication is initiated. And this origin of replication is collected from the PMB1 vector, and also this modification of the origin of replication is converted into the high copy number plasmid. High copy number plasmid. The pUC18 is a high copy number plasmid. As I told you that 2 important features of the cloning vector, that is a small size, which enables the cloning of bigger DNA fragments, and the second one is high copy number, so that the cloned DNA fragment is amplified more times, massive amplification, so that you can harvest and you can purify the plasmid and you cut down and you can use the various purposes. So here the pUC18 is a high copy number plasmid. Okay. Then in the next slide, I'm going to speak to you again the same features in the text format, right? Advantages of pUC18 over PBR322. High copy number plasmid, as I told you right now, 1500 copies per cell. Imagine 1500, okay, 18 plasmid per cell. That's high copy. And you see, this is because of small size. Because of small size, it can enter into the plasmid very easily, so that transformation rate is also high. So you get the more transformations, small size, and the transformation efficiency is more. Small size vector can accommodate the DNA fragments of 10 kb. As I told you that plasmids are stable up to 10 kb. Since it is 2686 into around 2.5, so you can clone another 7 and 7.5 kb larger DNA fragments. You want to clone larger DNA fragments, which are in size of 7 to 7.5 maximum, you can use this vector, right? Right. Then multiple cloning site or polylinker provides the many restriction enzyme sites, so that vector can be used to clone variety of DNA fragments, and you can use the two sites for directional cloning. You will not lose any fragment, because they are in the space of almost 20 to 30 nucleotides range only, right? And directional cloning is possible and controls the orientation of your DNA fragment. And single step visual detection of recombinants. That's recombinants will be in the white color colonies, and non-recombinants will be in the blue color through the alpha complementation. When you clone your gene of interest into the alpha laxed prime region, and the alpha peptide, which is coming from that vector, will not associate with the omega region from the omega peptide from the host. Then what will happen? The recombinants will not have a functional beta galactic space, and which cannot metabolize the X-gal, and recombinants will be in a blue color. And non-recombinants means where there is no insert in the laxed prime region, which encodes the usually alpha peptide. That alpha peptide can bind with the omega peptide of host and forms the functional beta galactosidase, and the non-recombinants or just transformants can hydrolyze the X-gal, that's a substrate, to convert into blue color. So visual detection of non-recombinants and recombinants is possible with the pUC18 vector. So in the next lecture, I guess I'll speak exclusively on what is alpha complementation. I don't want to speak right now. In the next video, I'll speak, but try to see that also. And next one is functional expression of the cloned gene is possible with the inducible promoter. So functional expression means it will not encode the high amount of protein, but it is possible to see the functional expression. Means you can assay that particular activity of the protein. If it is enzyme, you can see the enzyme activity from the recombinant one. If it is a transporter, it accumulates that substrate. Functional, not like a massive production of a protein, is not possible from this factor. So this is all about pUC18 elements and advantages of pUC18. So if at all you like it, try to subscribe my channel. That is Dr. K. PREM-PRIMER, right? And if you like it more, if you feel it is more useful, and try to forward to your friends. And if you have any comment, please let me know through the comment section. I'll get back to you quickly once I see them. Thank you all for listening. See you again with one more video. Until then, bye-bye.